close your microphone.
Hello everyone, thanks to stay here. And we'll start now. The objective of the discussed methods is to study the hominin adaptation to new climate events and landscapes through the changes of their diet. These changes are studied by the researchers of diet reconstruction. First, such studies focuses on animal teats because they are preserved the best. In addition to that, micro scratches on teeth can provide information about food consumed by the animals or the hominids and the use of their teeth and so far and so on so far. Nowadays, we have more methods to study the hominid diet, such as Macrowear, isotopic analysis, DNA analysis, or enamel thickness analysis. Now we gonna give you a general view of each of them with my colleagues.
Okay, so first let's focus on Macrowear analysis. So we will see contribution of Macrowear studies in general. So it gives us information about diet, about age at death, about demography, life history, food processing methods, and other cultural activities. So now how Macrowear can lead us to understand diets. So mastication of different food items lets identifiable traces, and these traces accumulate during the lifespan of an individual, reflects diets over a long period. So we can use for that the OFA method. OFA means occlusal fingerprints analysis. This method was recently used to analyze relationship between dental morphology and tooth wear pattern in maxillary first molar of Neanderthal and Homo sapiens. You can change, thank you. So we will see a OFA method step by step. First, dental replicas are molded using silicone, like what dentists used, directly on teeth. Then they fill the negative model with gypsum, and that gives us the three-dimensional model. You can change the slide, please. Thank you. And then 3D digital model is generated using white light scanning system, and occlusal surface of teeth can be quantified. And then diets can be identified based on what we know on physical properties of meats or plants, and after some statistical analysis. So now we can see micro texture analysis with Asna.
Thank you so much. Good morning, everyone. Macrowear texture analysis helps to reconstruct the aspect of diet among hominins. The limits, it reflects the difference in food preferences, which depends on the landscape. We can see here in this photo explains the type of diet. For example, we can MTA can identify the type of food if it is abrasive or non-abrasive. For example, nuts mostly responsible for abrading the enamel surface, unlike leaves or meat diets that go the opposite. We can also understand from MTA some ethnographic factors, like some hominins ingested hard particles that attached to their food as a result of food preparation technique that could reflect a highly aggressive diet. Unlike Macrowear, MTA gives individual diet in a short time, weeks or months prior to death. The advantages of MTA is that it provides us with a 3D data, a more realistic representation of the dental surface, so it will be easy to detect any small changes due to the combination of the white light confocal microscopy with the scale-sensitive fractal analysis that based on the Enamel surface can look different on when observed at different scale. For example, in the second photo, we can say the image A surface may appear smooth when observed at a coarse scale, while B and C can appear rough at finer scale. We can see this image explains the complexity, the changes, which is one of the parameters of MTA. So we can see from A to C will be called the calculated surface area at a particular scale. And then they will be defined, divided in the polynomimetric area, which is figure D. And then in E, we can see that the area scale fractal complexity will represent the slope of the steepest part of the curve that will fit into a relative area over the scale. One of the advantages also of MTA is that it is not destructive process, and also the observer error is eliminated with the help of the computer analyze, and the same values for the variable described generated each time the analyze is run on a given surface. Could you change the slide, please? We have 5 variables to describe the surface texture attributes. First, complexity, which is calculated change in surface roughness with the change of scale, reflect mainly for hardness. And then anisotropy, which is to measure surface texture orientation, and then the texture field volume, which is the measure of size and depth of the wear feature. And then we have scale of maximum complexity, which is the scale of increasing roughness that depends on the size of abrasive particles. And last, heterogeneity, which is a variability in complexity across the surface. And that's related to the variation in fluid properties. We can say in this image, the Macrowear texture schematic view showing high and low anisotropy and complexity and heterogeneity. We can see, for example, in the second part of the image, in the high especially, the anisotropic have a characteristic of the similar orientation, as all of them have a one parallel striation, and then the complex have a surface as heavy bits and scratches different overlaying each other. And then the heterogeneity, we can see the different in texture from place to place. And this is important because when we do comparison between species, we can see one parameter could be higher than the other. So it could give us a meaning of a changing of component of specific diet, or they can consume one type of food more than the other. Okay, could you change the slide, please? How to apply this method to prepare high-resolution dental cast to clean the teeth with cotton swab soaked in distilled water in acetone or ethyl alcohol. And then the positive cats will be poured using IMQP technology 301 resin. And then using the sensor configure image profiler, as I said before, it helps to see any kind of changes and also resolve the Macrowear scale details in 3D. And finally, the scans will be analyzed by the tooth fracs, the surf card, to generate the 5 variables that I mentioned before to describe the different attributes of surface texture. And next slides will be presented by my friend Boden. Thank you so much. Boden, it's your turn now.
Boden, your microphone is not on. Boden, we cannot hear you. Your microphone.
Last call for Boden.
Boden, we can see just your new hair, like a Mohican. At all, because your microphone.
Okay,
okay, it's okay now. Thank you.
Yes, sorry. So, stable isotope analysis study isotope composition of organic remains. It is always applied to organogenic chemical elements carbon, nitrogen, oxygen, and sulfur. It is important because these elements take active part in the body's metabolism, so their distribution depends on nutrition. However, here we will speak only about stable isotopes of carbon and nitrogen because they are the easiest to use. Expression delta C13, for example, means relation between relative concentration of C13 isotope in a sample and one in the standard realism of nitrogen is characterized first of all by the continuous concentration of its heavy stable isotope N15 in bodily fluids and proteins. Therefore, in food webs, there is stepwise trophic shift of delta N15 equal +2 to +6 promille because predators consume reveals two main aspects of diet. The first of them is position of the individual in the food web, proportion of meat consumed. The more meat one ate, the higher is their delta N15 value. Also, if one ate meat of predators, non-herbivores, it will be higher. Another aspect is amount of aquatic food. The thing is that aquatic food webs are much longer. Even plankton, which is always on the basis of these food webs, can be some small plankton organisms, can be some small predators. And so until proteins in these food chains reach fish large enough to be caught with paleolithic harpoons, they arrive to delta N15 value higher than in terrestrial carnivorous mammals. Delta C13 provides different kind of information. Stepwise shift is not pronounced here. Instead, distribution of carbon-stable isotopes differs in C3 and C4 plants. Specific formation in C4 plants, called bundle-Sches cells, fix and transfer carbon dioxide to other photosynthesizing cells. Thus, they maintain isotope composition of carbon. C3 plants, which are majority, fix carbon dioxide in traditional way with underrepresentation of C13 isotope in the produced organic substances. So if individual plants are typical for arid climate, greater for certain plants like maize, millet, sorghum, it has higher delta C13. Another aspect is fresh water or marine source of aquatic food. Algae commonly is mineral bicarbonate. In general, stable isotope analyses are quite precise. They often allow calculating approximate portion of different kinds of food in the diet, like portion of meat and plant food in case of N15. They allow identification of some more precise categories like fresh water and marine aquatic food. Finally, with assistance of other methods, we can even bring precise this. It is based on the use of polymerase chain reaction, PCR, with following computer aligning of the sequences and multiple complex comparisons with already known ones. Of course, it allows exact identifying species of plants and animals, thus being extremely precise method. It can be used in various ways. In an article recommended for us by Julie, authors identified diet of Neanderthals having fragments their oral microflora microflora's genome. So they supposed hominins with similar diet have similar microflora and inferred this diet. Important information here is identified species of animals eaten by Neanderthals, which is indisputable evidence of consumption of this species. Another study we found and analyzed revealed diet from coprolite DNA of humans, also finding exact species of ingested organisms. Here we can see difference in the peculiarities of obtained information. While fecal DNA gives us idea about plants ingested together in one meal, DNA trapped in dental calculus shows foods eaten throughout the life of an individual. In the end, to study gut microflora using DNA analysis, because it also can depend on diet. To conclude, we can say that DNA is very precise method which can work in different timescale frameworks, but it has one big problem. Poor, not contaminated DNA can be extracted from ancient bones very rarely, and from ancient dental calculus too. And always when it is extracted, we can rely in our analysis only on naturally numerous repeats of DNA, bacterial genomes, mitochondrial and chloroplast DNA, but analyzing of nuclear DNA is nuclear DNA of eukaryotes is much more difficult. And then I pass presentation to Maima with your analysis.
Okay, so another method that can be used to infer diet is the study of morphological traits such as enamel thickness. The methodologies used in this type of research are mostly microtomography, which is a non-destructive and non-invasive scanning technique that produces cross-sections of the objects studied in order to form 3D models. A recent study by Skinner and colleagues in 2015 analyzed enamel thickness in a large sample of Pleistocene fossil hominins using 2D mesial plans from microtomography. They examined the average and relative enamel thickness and distribution of the enamel of mandibular molars. The data used in previous studies consisted of values from linear measurements on damaged surfaces of molars. So previous research done on enamel thickness resulted in data with limited sample size and used linear measurements that resulted in a poor assessment of enamel thickness. The location of the measures also varied in different studies, which resulted in a poor assessment of enamel thickness variation for mandibular molars in Pleistocene hominins. In this study, the authors tried to control the variables and create a homogeneous set of data using microtomography and controlled mesial plans of section in mandibular molars. So in this figure, we can see an illustration of the protocol used to collect enamel thickness data. In the top left corner, we can see a surface model of a mandibular first molar with a red line indicating the location of the 2D plane of section. The top right corner shows the 2D plane of section for this specimen upon which measurements were collected. Bottom right shows the surface area of the enamel cap in yellow and the dentine in blue. And bottom left shows the bicervical diameter measurements, red line, and the enamel dentine junction length measurements in a white line. While the black lines delimit the lingual, occlusal, and buccal regions used to measure the distribution of enamel across the crown. The methods used in this study allow comparison of data since the protocol they followed is repeatable. Previous study relied on enamel thickness data that were taken differently and followed different protocols. So the data was heterogeneous and did not allow for comparison. The new imaging and visualization techniques used in this study are non-invasive and allowed the study of variation of enamel thickness. So to assess the ability of this method in reconstructing dietary adaptations, we know that the consumption of hard and abrasive grass-based foods is associated with thick enamel. When the results of this study were associated with results from other research on isotopic and microwear research, an increase of C4 consumption was demonstrated in Australopithecus annamensis, Australopithecus afarensis, Australopithecus aethiopicus, and Australopithecus boisei. The authors argue that this increase in C4 consumption is also associated with an increase in enamel thickness. So the results show a general trend of increasing enamel thickness for Australopithecus species during the Pliocene, with Australopithecus boisei showing the thickest enamel. We conclude that the methods used in this paper can be used as a valuable tool in reconstructing diets based on enamel thickness and allow for comparisons between taxa.
As you could see, we present 4 different methods to study the hominid diet. All of them can provide different and specific information. Because of that, they do not exclude each other. We can study the short term with Macrowear studies, or a long term with Macrowear or DNA analysis, the current food with the isotope analysis, and so on and so far. To some, it's necessary to combine all these methods to get a complete picture of the hominid diet and to be able to verify the obtained information with all of them. Change, please, the slides. With that, thank you for your attention. That's all.
Okay, thank you very much for your presentation. Thanks also to have respected the assigned time and to have organized well your presentation that was very clear. So I ask to my colleagues if they have questions or remark. Who wants to start?
I can start.
Please, Julie.
Okay, thank you very much. You respect the time, as Marta said, and your presentation was clear, and we see that you have tried to do something all together. So congratulations for that. I have not a lot of questions. I have just some curiosity of mine, because I saw in the PowerPoint that concerning Macrowear analysis, you talk about the possibility to infer some cultural activities. So my question is, which kind of cultural activities you can infer from the Macrowear analysis, and how you can make the differences between cultural activities and food nutrition? Maybe it's a difficult question, but it's only curiosity if it was mentioned in the paper. And correlated to this question is if these cultural activities can also be infer from the Macrowear analysis?
Can I speak in French to answer?
For me, it's okay, of course.
Ok. J'ai lu dans le papier que apparemment, par exemple, pour fabriquer des paniers en osier ou avec des tiges, on pouvait le remarquer sur les dents lorsque, par exemple, il mordait pour casser la tige ou des choses comme ça. Si c'est une activité qui est assez répétée, on peut le voir avec ce genre d'analyse, quoi. J'avais lu ça, mais voilà.
Ok, non. Très bien, merci. And about Macrowear, do you read something about possible traces of cultural analysis from Macrowear, Asna? Maybe not.
I read the article, Serena Dietary, that mentions that some of the she was making comparison between Neanderthal behavior and modern human and also Paleolithic group. She mentions that sometimes it depends on the context of the samples. If sometimes they used technology, technology could sometimes help to change the static of the food. If it was hard food, it could be using some kind of technology that soften, change its morphology. This is what I could understand, that it was also culture, like the technology could help to understand the type of food changes. I hope it could cover the question.
Okay, thank you.
Thank you.